Hello everyone, in this video I'm going to share about experimental design in research method. So what is experimental design? It is another type of a scientific approach that we use to solve our answers in a research method. So we usually come up with research questions and we need a direction on how we want to go about answering the question. So experimental design is one of the ways that we can use to answer our research questions. So in experimental specifically, what we would like to examine is the effect of one variable to another. It can be one independent variable and it is affect to one dependent variable or multiple independent variables and the effect to multiple outcome or dependent variable. But what we are trying to see here in experiment is how much does one variable is affecting another. So that can be only captured from experiments. So if your objective is to find, or your research question is to find an effect of one variable on the other, then use a experimental design. So that is what we call as testing causal effect. So does one event or one variable is causing an effect on another variable? And this method is very widely used in social sciences, psychology, education, and medicine. So all this while we are talking about COVID-19. So there are a lot of experiments are being conducted. So a lot of new drugs are tested on the patient to see the effect of these drugs on curing the person, curing the patients. So do they cure the entire disease? No, they would like to see how does one drug can reduce a fever. How can the drug reduce the respiratory system? So it is a causal effect studies. And a lot of time experimental, we always think about laboratory test procedures because it is very rigorous procedure. You have to follow very strict guidelines when you're doing experimental because you want to answer a question or you want to find the causal effect. So when you want to say one is affecting another, how sure are you that this independent variable is the one that is causing the change? Going back to the COVID-19 example, how to assure that this medicine is actually helping to reduce the fever or reduce the symptoms for COVID-19? So you need to monitor the patients. You have to make sure that nothing else is making the person feeling better or curing except for the drug. So you have to ensure every other thing is being hold constant or being put it on a constant state, like nothing else is affecting the recovery process except for the drug. Only then you can say that the drug has made an effect and made a change in the recovery process. If not, it could be other reasons. Maybe the immunity itself on the person's body could have caused a recovery. Maybe the belief system in that patient could have helped him cure or recover from COVID-19. So there are a lot of other factors. So you need to control all these factors and then you administer the drug. And when everything is under control, you can easily see if the recovery is because of the drugs or because of other factors. So if you want to make it even easier, this is like an example of doing experiments in the lab. Okay, so when you mix chemicals in the laboratory, you know why you get a blue color, because you mix a red and an orange, then you get a blue. So, and no other factors that contributes. And another example is in biology, doing testing on drugs using mice or lab rats. Okay, so these lab rats, why do we use lab rats? Because they are being grown, or they are being fed with the correct materials. They are being monitored, they look the same. We don't take regular rats or mice to do any lab testing, because they have different factors that might bring in. They are different. Lab rats, the rats that you breed inside a laboratory, is different than the regular house mice or the one that you can get from the drains. So you cannot assume that the drug works because of the drug itself. It could be other reasons. So in order to eliminate all the possible factors contributing to the outcome, you need to conduct an experiment. So what is the features of experimental study? So when you're talking about experiment study, these three things are always the clues or indication that this is an experiment. Or if you want to do an experiment, these are the things that you need to consider, the very important elements. Firstly is the variable. So you need to have two types of variables. One is independent and the other one is dependent. So independent is actually the factor that brings or causes any change to another factor. So this independent variable is the variable that we manipulate, okay, in the study, so that it can affect another variable. And the dependent variable is the outcome. So we manipulate the one independent variable, and then we see how does this independent variable affects the outcome variable. So this outcome variable is what we call dependent variable. It can be measured at one point, or it can be measured multiple time points. So for example, if you are drinking body slimming liquids or supplements, so you cannot expect changes in your body within the next day or within few hours. So you need to keep record of the changes for at least a month. Only then you lose weight because of the slimming drink, or is it because other factors? Were you on diet, or were you having depression? That's why you lose weight. So that is what we mean by measuring the variable or the outcome over a time of period. Okay, the second one is random assignment. So in experimental study, we will give our participant equal chances to be in a treatment group or the group without treatment, or we have equal opportunity to be assigned in any groups. So it's not necessarily always treatment and control, but this is a very, very basic example of groups that you can assign people into. Okay, because remember, you want to identify the cause and effect relationship. So if you want to know the cause and effect, you need to have comparisons. You have to make comparisons between groups. So in order to make comparison, what will make them different? One should get the treatment or intervention, the other one will not get it, and then you can make comparison between them. Okay, now the third element is groups. So I think we have talked about it earlier. Before this, you need to have at least two types of groups when you are doing experimental study. One is the group that is exposed to the treatment or experiment or intervention. The other group that shares the same experiment environment, but it will not get any treatment or intervention that you would like to examine. So very simple example is there are patients for COVID-19 who will get the real drug to examine the effect of the drugs, and others are also in the hospital setting but will not get the treatment drug. They might be under regular treatments, so that they can make comparison between the patient who received a new drug in their treatment and the other group who did not receive any treatment or drugs. Okay, they are getting regular treatments but not additional drugs. So what are the example of experimental research? What are the type of experimental research that exists? Like survey, we have 3 different designs. So for experimental, how can we conduct this research? It can be through completely randomized design, can be through randomized block design, or it can be matched pairs. So let's look at the first design called completely randomized design. So this design is the most simplest design to use among other experimental designs, and this experimental design is used when you have a sample or population that are homogeneous, that are similar to each other. So meaning the sample that you are taking has similar characteristics or going through the same environment or living in the same environment, experiencing the same environment. So your population generally are the homogeneous. Homogeneous means same. So these experimental units are known as participants. So remember, when we are learning sampling, we use the term units. So units are participants. So in here we are using experimental units because we want to highlight that this is an experiment design. So when we want to do the sampling, we are taking it from a pool of similar peoples going through the similar conditions. Okay, and it is very well adapted in experiments with small number of treatments. So it is very applicable when you have like 2 or 3 different type of treatments. It will not be applicable you have like 10 different treatments and then you want to do a completely randomized design, because you need to have large sample who are similar. Okay, because initially we said we want to have similar samples. So if you have a lot of treatments, you need a large group of people who are similar to assign them. So it is not applicable. So we usually use this design when you have smaller number of treatments. And then experimental or the participants are randomly assigned into treatment or not receiving treatment, and both of them has equal probability to be in either group. That means every individual in the sample that we have selected has chances to be in treatment or control group. So it is like they have equal chances, like 50-50 chances to be in treatment or control group. And randomization, when you randomly assign them into treatment or control group, they are being assigned using random number table or computer programs. Okay, now let's look at the steps. Firstly is you need to identify your sampling frame. This is a step from sampling procedures that we learned few weeks back. So using the probability sampling. Okay, why do we use probability sampling? Because this is an experimental design. We want to ensure that every individual has equal probabilities. Okay, so once you have your sample, okay, once you've selected your sampling frame, you have to take your potential sample out of it. So what we do is we assign numbers to the list of the participants, or in this case we call experimental units, and then we take all these numbers and put them into a a box and we draw out the numbers, draw out participants into treatment or control group. Okay, so in a pic we have a list of participants, we assign them numbers, and then we put all these numbers into a box, and then we pick one at a time, and the first one goes into treatment, and you pick the second one, and that participant goes into the control group until you are done with the assignments. So until you are done with all the numbers you have in the board, or when you reach your required sample size. So when how do you decide whether they want to go into treatment or control? What I did was a very traditional way. You write all the list and you put it in the bowl and you just pick randomly, and that is not something that you will do if you need 200 participants or more. So what do? We use random number generator, or we use the random number table. Then from there we assign them to treatment or control group. In the treatment group, they will receive a specific treatment or drug, and then we measure the outcome. But in control, we only measure the outcome. We do not give them any treatment. That's why that was what makes the two groups different. And once you are done measuring the outcome, we make comparison between the outcome from treatment and control. So let's say, for example, you would like to study the effect of a drug, the HIV drugs, on the COVID-19 patients. Okay, so what we have is we have a sampling frame. So we have all the patients in Malaysia. They are the accessible patients for it. So what we can do is we can get the list of all the patients. We can assign numbers to each of the patients, just like what the government has mentioned, right? So if you notice, sometimes even in the press meet, the health director mentioned case 120 is who and who passed away when and when. So actually all of them has been assigned an ID. So all these IDs will be collected and put into one box. And then as a researcher, what you do is you select one and then put it into a treatment group, and then you select the second patient from the pool of patients and then put them into control group. So you do it until you reach your sample size. So if you say that you need 50 and you need 25 in each group, so you need to draw 50 times. Okay, so alternately you put them into treatment and control. So just like as I said, so you have pool of patients. Okay, you draw out the first patient and you put them into treatment. Then you go back to the box, you take another number or patient's ID, you put it into control, and you do this until you reach your sample size of 50. And then you take these patients who are in the treatment group, and then you give them these HIV drugs. For the control group, you don't give them any drugs. And after few weeks, let's say that after 1 week, you measure the outcome. For example, here you measure the level of fever, for example, or you measure the recovery process or reaction to the medication. So you measure the outcome for both the treatment and control group, and then you make comparison between these two groups outcome. Like I said, you measure, for example, you measure the fever for all these patients in treatment group after 1 week of given the HIV drugs. After 1 week, without giving any drugs, you measure the control group's fever or temperature, and then these scores from treatment and control group will be compared. And when you compare, you're able to see if there is any differences in the fever values between treatment and control group. If there is, that means the drug does have an impact. Okay, so now we are going to talk about the second type of experimental design called randomized block design. So this is different from completely block design, and this is a little bit complex compared to the regular completely randomized design. So what makes it complex? Okay, it uses the concept of blocks. Okay, so these experimental units or the participants are being grouped into blocks or variables that could also contribute to the outcome. Okay, and within each block the conditions are homogeneous, meaning within the block the participants are similar, they have similar characteristics. Okay, so to understand better, let's go back to the let me explain the procedure, and using the same example as COVID-19 example that we talked about in completely randomized design. The first steps are similar to completely randomized design. You have to identify your sampling frame, you use probability sampling, and you assign them with the numbers or ID. Okay, so what you do next is you identify, you use random number participants to identify experiment or the experimental units into the study. So when you already assign numbers, you have to select them. Okay, select your participants. So once you select the participants, you don't assign them into treatment and control yet. You just take the sample. What you do next is you have to block them, you have to group them. Okay, and meaning that you are actually splitting the sample into blocks. So within the block you assign the participants into treatment and control group. Okay, let me explain using this COVID-19 example. So let's say that you want to examine the effect of HIV drugs on the recovery of COVID-19 patients. So you want to examine whether this HIV drugs can help to recover, help recovery of these COVID-19 patients. So what you do is you take all the samples that is accessible to you in Malaysia, and then you list them out. They've already given identity to each of the patients. So what you do is you collect all this identity and put into a jar or a box. Then or you use random number generator and you select the sample size that you want. So from 1,000 over patients, maybe you select up to 400 patients. Okay, so and this is randomly, you select your 400 patients, and then from these 400 patients, what you do is you split them into male and female groups. Okay, so you split them by female and male groups. So that is what we mean by block. So gender is a block. So what you're trying to do is you're partitioning the samples, okay, to help us further explain the effect of the drugs. Because gender, the way male and female reacts to a drug, can also cause differences in the recovery process of COVID-19 patients. So maybe female has more tolerance, or male has a better tolerance level. So that could be another factor for recovery process. So what we are trying to do is we want to explain very clearly the drug is actually causing the changes in COVID-19 patients, not because of whether they are male or female. So how do we control for this male or female elements? So you block them. Okay, so you block them. So very first stage is you put in gender as a factor. So from the sample of 400, you select all the males into block 1, all the females into block 2. And within the block 1, within all the males, what you do is you randomly assign them into treatment or control group. Same applies in the female block. Within female block, you assign them into treatment or control group. And then you those in the treatment groups for male and for female will receive the HIV drugs. So after 1 week, the outcome, okay, for example, the recovery process or the fever of these patients, the male patients under treatment, the male patient without any treatment will be measured. Same goes to the second block. You measure the temperature or the fever of female COVID-19 patients who have received HIV drugs and those who did not receive any drugs. And you make comparison between the treatment group and control group within this particular gender. So you see, is there any differences in the fever of COVID-19 patients after taking HIV drugs for the female or for the male? So you are very specific on the looking at the differences for specific gender. Okay. Lastly is the matched pair design. Okay, this is also different compared to the others, and this is even adding more complexity to it. Okay, what makes them different is the way you select your participants. So all this while the experiment, you are still making comparison between treatment and control groups, but how do you select them makes a big difference. So here we are calling it as matched pair design, meaning we are actually getting the experimental units or participants to be paired first before assigning them into treatment or control. Okay, so what we are trying to say is you will want to find a group that is as similar as possible for treatment and control group. Okay, so it's like finding a twin, a pair of twins for this experiment, so that one person, one member of the twin will be in control, the other member can be in treatment, because both of them has similar characteristics, similar images or similar factors. Okay, similarities between them, they are as homogeneous as very homogeneous, but they are not identical. You cannot get the same participant in both groups. Okay, because you are doing the study simultaneously. How can a person be in the treatment and at the same time in control? It's impossible. So what we try to do here is we try to find somebody who has as similar as you are, as identical as you are, into another group, so that they become like your replica or your twin in the other group. Okay, so what they do in matched pair design is, once you get a sample, let's say that you got from your sampling frame, you do a probability sampling, you gather about 400 individuals. So from these 400 individuals, they try to find pairs. Okay, so for every experimental unit, there should be a pair. So you match them first. So if it is 400, then it turns out to be 200, because there will be 200 pairs in your data. So for each, these pairs can be matched using key variables, like for example age or IQ scores or height. Why? Because when you have the same age, the expectation is you will have similar maturity, similar experiences. So as I said, we cannot find somebody exactly the same, even there is differences between the twins. So we want to find somebody as similar as to the other experimental unit. So we want to find similar people so that we can identify the true cause that is actually happening in the study. Okay. So by having similar characteristics, we have high homogeneity, and we have to remember matched pair design only applies for studies with two groups only. Okay, so as I said, COVID-19 patients. Okay, let's look at the same example. Let's say that we wanna test out if an HIV drug is helping in the recovery process of COVID-19 cases. So what do? We have the entire list of accessible COVID-19 patients, and then from there we take a sample of, let's say, 400 participants, 400 patients, and these 400 patients, for every patient we try to find the best match. So how do you find the best match? Okay, if you notice, we might have patient with similar medical history. Okay, if you've been following the news on COVID-19 patients, there are a lot of similarities between the patients. They have chronic illnesses, and they have similar chronic illnesses like high blood pressure, they have diabetes, they have heart disease, and they have age range that is similar, right? Most of the patients were 60 and above. So all these are the key variables that you can use to match a pair. So you can use maybe just age, or you can find somebody with a combination of age and chronic illness. Then you use that to find a match. So for example, patient A, A001, the ID, this patient is a male with chronic heart disease. So in that same sample, in that 400 participants, what you do is you find a person who is a male with chronic heart disease. So you will have a pair of patients who has similar age and medical condition. Okay, so you do for the second one until you get 200 pairs. Okay, in some cases you will not get the exact you will not get a pair with meeting the exact condition. Sometimes you will get male or 67 years old, but there is no more potential options, so you might get a female who are 67. So but you try to get the best match, the most similar. As we said, we cannot get identical matches, so you have to find as similar as possible. Okay, so once you get a list of pairs, okay, so let's say you have a pair of AC, a pair of B and E. Okay, so what you do next is you assign one of the member in the pair into treatment and the other one into control randomly. So you say, okay, now I have pair 1, I take A into treatment, B into A into treatment and C into control. And my second pair is a combination of individual B and individual E. So I assign the individual E into treatment and individual B into control. So end of the day I'll have two groups, treatment and control. In treatment I'll have members of A, E, and in control I'll have member of C and B. So this treatment group consists of A and E will receive HIV drugs, and then after few weeks we will measure their fever or temperature, and then on the same week we will measure the temperature and the fever of the control group. So the scores or the values that we get from the treatment group and the control group for the fever or the temperature will be compared to determine whether there is any differences between the values. If there is a differences, we would like to see, is it caused by the drug or otherwise. Okay, so in this matched pair design it will give you much more rigorous or much more confident results, because the members in the group are similar. It is like you are having a mirror to another group. So if you are in treatment, you have somebody who is a replicate of you in control. So ideally, if I don't receive treatment, what is the outcome that I might get if I'm in control? Okay, so what will be my temperature reading if I'm not receiving any treatment after 3 weeks? Okay, in comparison to it, if I'm in the treatment group, I'm receiving treatment for 3 weeks and I record my fever or my temperature after 3 weeks, and what will the results be? Okay, so I can make comparison. It is making comparison of the same individual. It's like doing a matched or dependent t-test. You're having the similar sample tested twice, but even though we say similar samples, the samples are not identical. Okay, they still have some variation, because we are human. You cannot get exactly identical people in both groups. Okay, so to understand better about experimental designs, think back or review back all the information on COVID-19. Okay.